We're in the Pacific, about 1,000 kilometers west of South America, on the equator. Martin Wikelski is heading for his research site. It's an island called Santa Fe, part of the Galapagos archipelago. Santa Fe, like all the Galapagos Islands, is the tip of a volcano that became land only a few million years ago. Many of the animals and plants that now live there are found nowhere else on Earth. These island species have long fascinated biologists interested in evolution. But this is also a good place for animal physiologists to study. Like all animals found in isolated oceanic island groups, the species found in Galapagos are astonishingly unafraid of people because of the absence of predators. And even on an inhabited island, on a hotel patio, marine iguanas, a Galapagos species, lounge in the shade of the chairs. With few natural predators, they don't see people as a threat. They're easy to observe and study, and a source of fascination. Martin's work is on the marine iguana. He's going to Santa Fe because it's home to more than 10% of the world's population of these animals. And here, they're undisturbed by humans. If you study the animals here, you're as close to understanding them as you can get. This is the nitty-gritty of research. What we can read in textbooks is knowledge won from situations like this. There's actually an easier way onto the island. There's a beach. But it's 4 kilometers away, and that would mean carrying all the equipment to the research site. So the researchers prefer the quick route. Most of the islands in the Galapagos archipelago are uninhabited. In fact, you need permission to land on them. Everywhere the human species goes, it affects the environment. This is one of the few remaining places on the planet where human influence and access is being tightly controlled. It took Martin months to secure permission to work here. More people have been up Mount Everest than have been allowed to come to this place. This will be the group's laboratory, eating and sleeping quarters. Everything needed for the 3-month stay on the island has to be brought with them. And it's a condition of their permission that everything is taken away at the end. So now they're here, what are they going to do? First, catch your animal. The iguanas don't fear him, but are slightly wary, staying beyond arm's length. Again, this remarkable lack of fear of humans. It doesn't like being handled, but it is astonishingly passive. The basic data first. Body temperature, obtained by putting a thermometer into the cloacal pouch, which all reptile species, males and females, have.
It's 30.0.
For a cold-blooded animal, the temperature may seem high, close to mammalian blood heat, but it has spent most of the day soaking up the sun. Next, length and weight. These are basic pieces of information, but are crucial in understanding how this creature lives.
Es 2 kilos y 100.
That's 2 kilos and 100 grams. And for the next 3 months, this animal is one of the sample that are going to be intensively scrutinized. So it needs to be picked out from the crowd on the rocks. Each animal is given a distinctive mark and number. The range of species found here, and here alone, makes these islands an endless source of fascination for biologists. Santa Cruz, the main inhabited island in the archipelago, is home to most of the scientific work. At the Charles Darwin Research Station, a study is underway which relies as much on the geographical position of the Galapagos, on the equator, as the species itself, the giant tortoise. Beatrix Schramm collects environmental information. First thing every morning, she measures temperature, humidity, and takes a reading of light intensity. She's interested in the mating behaviour of the tortoises and is trying to find out what triggers it. As the Galapagos are on the equator, the length of day doesn't change all year round. So what is triggering the mating behaviour? Beatrix is looking for measurable signs of sexual activity. For the physiologist, the starting point is sex hormones, which can be found in the urine. Collecting urine from a 70-kilogram tortoise is a bit of a problem, so she gets them from the next best thing: fresh feces.
Yeah, this fecal samples, I'm not sure from where, and this other one there too, I'm not sure from whom it is, because as you see, this tortoise is just running away now, and because they are so fast, it could also be from another one. I mean, in this morning, I don't know who else stayed here. So this will be a little bit elder because it's not so humid anymore. This perhaps, yeah, a little bit elder, like 1 hour or something like that. You can check this. If there is lots of humidity, they just made it. And that's why I have to run. If I see a tortoise and she's sitting somewhere and she also saw me, they start running. So I have to run too, to look at from which direction she was coming. And when I just saw, yeah, she's running exactly away from this fecal sample, which I could see now, and she's just like half a meter away. I know this is from her. But the best way is always that you can see the animal sitting exactly on the top of the fecal sample, that you can see where the tail is, there is some feces, and you can collect it. That's the best.
The animals produce the feces first thing in the morning, and she needs it fresh. They know she's around, so it's a bit of a game of hide and seek. Even when the animals are secure in a rock-walled compound, finding them can be tricky.
Beneath.
Tortoises can be extremely quick and are very shy. To use a sample of the faeces in her research, she has to be absolutely sure that it has come from the tortoise she has spotted.
Let's see, there is one. There is one.
And the sample really must be fresh. Any delay and the hormones in the faeces are degraded very quickly in the heat.
I know exactly where she's sitting. So if she's running away when we're coming, I know that this is the feces of her. And now, it's a very fresh one. I have to collect that.
Success. The next job is to stabilise it before any changes take place. In a nearby lab, she teases out a 4-gram sample of the material and suspends it in alcohol. The sample is now ready for analysis. But although the station has sophisticated facilities, compared to Martin Wikelski's iguana research setup on Santa Fe, we're still over 1,000 kilometres from the nearest equipment capable of analysing for the testosterone and oestrogen at the extremely low concentrations found in faecal samples. So the samples are chilled and stored for later analysis. The telltale signs of the volcanic origins of Santa Fe are littered all around. For most of the year, the island is parched, desert-like. 3 months can seem a long time in this sort of environment. It's the sort of research you need to pace yourself for. The marine iguana is something of a curiosity. It's the only iguana in the world that lives by and in the sea, and it's only found on the Galapagos archipelago. Iguanas probably arrived on these remote islands on rafts of vegetation and adapted to the local conditions. Some, like the land iguana on Santa Fe, adapted to eating the cactus pads. The marine iguana has adapted to eating the seaweeds on the rocks close to the shore. But foraging for this food has caused them a number of problems. As they are cold-blooded and the sea is cold, they must warm up in the sun before going down to the sea to graze on the algae. The chilling effect of the sea means they must rest up for the day, warming up again, passively digesting their food, with the males indulging in the occasional bit of territorial skirmishing, or having ticks picked off them by a ground finch. Until the next foraging excursion. The chilling effect of the sea is apparent in the effort that they have to make to get back onto the land. After a long excursion, holding onto the rough lava blocks and climbing to safety is visibly draining. But how draining? What is the measurable impact on the animal? With a simple strain gauge, the animal is pulled off the rock. Correlate this against its weight and body temperature at the time of the experiment, and you have an index of stamina.
So that's 5 kilos exactly.
But there's one problem with this experiment. If all the iguanas he can catch are hot from basking in the sun, how can he measure one as if it's just come out of the sea? Improvise. A nearby tidal pool, used as a playground by sea lions, is a convenient place to bring the iguana down to a low temperature. But you have to shift the residence for a while. Another index is speed. Time trials for iguanas. They take significantly longer periods to run the course when they're colder. It's as good an index of performance as you need. What's more, the experiment is conducted so close to where the animals live, the disruption to their lives, always a problem in experiments on animals, is minimised.
I will put it here if you can help us there. Okay.
Beatrix needs a check on whether the sex hormones collected from the faeces are a reliable indicator. She periodically takes a blood sample from her tortoises. This week, it is the turn of a large male called Chico, a procedure which I'm lucky enough to help with.
Numero 4 era? 40 y por eso he pensado, ¿dónde está el 4? Sometimes we have to change the arm because it's the same with us. Some veins are good on one side, some are very bad. Está cerca. Porque hay sangre aquí. So we try the other side. So you can see here this part and these lines going from there till this tendon. Here is a tendon from the muscle, and we are following this line going in this small hole. Here's the vena brachialis going up there, and so we try to find this vein. You can't just feel it, so you have to following these lines. That's the best. But nevertheless, sometimes it's difficult to find it, sometimes not at all. I got it, you see? When you got it, it's just a few seconds and then you have it. Muchas gracias, chico. Sí, por favor. Yeah. Now we have to be very fast. This is a heparin tube. Thank you very much. So this is a heparin tube, and afterwards I will centrifuge it. I will centrifuge it for about 5 to 10 minutes. We only need the blood plasma inside, so we'll throw the blood cells away. And in the blood plasma there are the sexual hormones, and these we need.
At very low spring tides, when rocks which are normally covered are exposed, the iguanas graze the red algae. Martin takes the chance to sample the weed, with a watchful eye on the waves. The samples are analysed to identify the species. Each has a particular energy content, and the growth rate of each is known. If you know what's in the animal's stomach, you get a picture of the energy balance, what and how much it is eaten. To an expert, the chewed fragments from the stomach are identifiable. The purple coloration comes from the seaweed itself. For a complete picture, fresh faeces are collected from the rocks, dried and analysed for energy content. Altogether, the researchers have a snapshot of the energy balance of the animal over a day. What it eats, how much it eats, what it loses, and as a result, how much energy the iguana has extracted from its food.
And now, cuidado. Yeah. Por favor, Camilo, ¿te puedes ir a la izquierda? No, que ahora le sacamos, ¿no? Solamente unos metros. Ahora lentamente. Acostado, ya. Muy bien. Poco más en mí. Ya, gracias. Ya. Poco así. Ok.
It's ultrasound. The same technique used to look at human babies in mothers' wombs.
Oh, mira cómo son grandes.
The state of development of the early stages of the eggs, the follicles, is a clear indication of where the animals are in their annual sexual cycle.
Now you can see here it's ending. You don't see this side so good, but nevertheless, I mean, you can imagine this is the follicle, growing follicle, yeah.
As they approach the time of readiness to mate, the follicles show up as healthy, viable. If they are not fertilized, they may be reabsorbed. Beatrix can see the differences between them. The ones that are breaking down, known as atritic follicles, have characteristic hollow-looking features.
Sometimes you can see, normally you can see a very black hole inside here, so you just see some different colors like blackish, grayish, and it's not, normal follicle is totally white. Atritic follicle has some different colors inside.
The Galapagos can seem like a last frontier. The difficult and sometimes primitive conditions in the islands can suggest a lack of sophistication in scientific research. But making devices like the ultrasound recorder work in these circumstances is nothing less than a research tour de force. For Martin and his group, their efforts lie in turning their shelter into a laboratory, where they can undertake the precise and meticulously detailed work of inserting radio transmitters under the skin of selected iguanas. Why go to all this trouble? If your only means of keeping track of the iguana is by looking at them, you won't know what they're up to when they're out of sight, or when they go into the sea. What's more, if you're trying to correlate the animal's activities with the information on food intake and body temperature, you need some quantitative measurement, some data. You need a monitoring device that travels with, or in this case in, the animal as it goes about its daily cycle. Once in place, the transmitter sends back signals to the researchers as a series of bleeps. These are counted off against the clock. After a few calculations, the researchers have a measure of the animal's body temperature throughout the day. They can start to piece together a little of its life.
You see here a file of time against body temperature, and you see immediately that with the telemetry we get some nice data sets on certain aspects, on certain time periods. But you see, just between say about 20 hours and in the morning at 6 hours, we can't continuously record the body temperature, and we just have to assume that there is a continuous line, a dropping body temperature. We have some nice information here on the time when the animal was foraging, and that's also some nice aspect of the telemetry, that we get immediately the data on the body temperature, and we can design experiments to change certain aspects of the body temperature.
As the graph shows, there are gaps. The technique is flawed. The radio signal is quite weak. If the animal goes behind rocks, the signal is blocked. You only get information while the researchers are around. Martin has started to use a new invention, a device which measures heart rate and body temperature once a minute, and stores it on a microchip sealed within the implant.
Al aire no me lo pones.
The whole device is sealed in a waterproof resin and inserted under the skin of an anaesthetized iguana. There it will stay for several weeks. After the iguana has had a few days to recover from the operation, it is released and the data collected. Later, it will be recaptured and the device recovered. Then the information can be analysed.
You can see that even though the temperature drops down in the night, there are some changes, and this is probably because the animal moved its position inside the rocks. And you see a really smooth warm-up curve. So we can calculate a mathematical function about this on this warm-up curve. And there's no inaccuracies in the data because people didn't count right or take the time wrong. And you also see that during the foraging period, there's a really nice drop, and then afterwards an increase in the body temperature again. Then also the nice drop in body temperature in the evening or in the afternoon, and a little bit of increase again when the animal moved from its rock to a rock that was slightly further in the sun. So that's what they do around 4 o'clock or so in the afternoon. So all this can be nicely picked up and interpreted into this file if we combine it with our behavioral observations.
Among other things, science is about taking your chances when you find them. Martin has always been interested in the diving physiology of the iguana. If he understands how the heart operates, he's got a clue about the efficiency of blood flow. Quite by chance, he finds out about Beatrix ultrasound work on the main island. He jumps at this chance to look at the dynamic of a heart operating. It's another piece of the scientific jigsaw in place for nothing.
So this, you can see the heart pumping here, and now we can make it like a little bit bigger. You see the movement, and here
Oh yeah, that's pretty
it's
nice.
the largest one, yeah. And here it's ending, and here too. If you move with the ultrasound, with the head of the ultrasound a little bit, and you can see how the blood is going through, the rhythm of the heart here.
What we've seen here is a snapshot of research taking place, questions being asked, puzzled over, and experiments set up to provide answers. Some of the answers are already there, but for the most part, it's a long, continuing process, slowly assembling pieces of information which, as a body of knowledge, informs our understanding of the natural world. And perhaps more importantly, allows us to conserve and manage this fragile fragment of the planet.